we want to cover is cell death. And basically the idea, before we actually jump into the topic, the idea to keep in mind is, so far we've talked about stress, and we said that when a cell or an organ is placed under stress, it may undergo growth adaptations. So for example, an increase in stress results in hyperplasia and hypertrophy, as we've already covered. A decrease in stress would result in atrophy. A change in stress would result in metaplasia. If the stress is severe, or if the cell doesn't have the ability to adapt, then injury will occur. And the initial stage of injury is reversible. Basically, it's called reversible cellular injury. And if the injury persists, the reversible injury can actually become irreversible. And that irreversible injury is also known as cell death. And so that's really where we are in our global sort of grand scheme view of things. Now, when we talk about cell death, the morphologic hallmark of cell death is important to be familiar with, and that basically is the loss of the nucleus. I.e., if I'm looking at a slide, and I'm looking at cells on a slide, if I see that the cells no longer have their nuclei, that indicates that the tissue is now dead. So the morphologic hallmark of cell death is loss of the nucleus. It's not enough to know that. The examiners like to go a step further, and they like you to know the mechanism by which the nucleus is lost. And the mechanism by which the nucleus is lost is basically threefold. First, the nucleus shrinks down, and that's called pycnosis. Actually, pycnosis, the word pycnosis means ink dot, and the nucleus basically shrinks down into a small little ink dot. After shrinking down, the next stage is breaking up of the nucleus, and that's called karyorrhexis, where the nucleus breaks up into big pieces. And finally, the final stage is karyolysis, where those pieces are then actually broken down into basic building blocks. And so the three phases of loss of the nucleus are pycnosis, karyorrhexis, and karyolysis. Very important to be familiar with, relatively high yield terms. Now, there are two mechanisms of cell death. The first is, and we have to be familiar with both, the first is necrosis and the second is apoptosis. And the way I kind of think about these is necrosis is sort of murder. Something comes from the outside, or some pathologic process from the outside occurs, which then causes a grand scale destruction of the tissue. And that's necrosis. And apoptosis, on the other hand, sort of the way I think about it, it's cellular suicide. The cells independently sort of decide to remove themselves from the organ. Usually involves single cells or small groups of cells, and often there is no extensive tissue reaction to apoptosis. These principles will be highlighted as we move forward in this presentation. So necrosis. Again, necrosis is death of a large group of cells. That's important. And very important is that it is followed by acute inflammation. This is going to come into play later as well when we cover inflammation, but I'd like to actually stop here and stress this point. There are two processes that result in acute inflammation. The first, and the most commonly sort of thought about, is when you get an infection. If you get a bacterial infection, you're obviously going to get acute inflammation. But the second driving factor for acute inflammation is actually necrosis. So for example, when patients get a myocardial infarction, one of the early changes that occurs is that neutrophils come into the tissue. And another important change is that the neutrophil count goes up in the blood because neutrophils are being generated in order to be sent to the tissue. So necrosis, I would say, is very important to remember that it is followed by acute inflammation. Now, necrosis is always pathologic. It's always due to some pathologic process. And it's never physiologic, meaning we don't have necrosis occurring as part of our normal physiology. It always means that there's some disease or problem present. And it can be divided into several types based on gross features. These are high yield. These questions often show up on examinations concerning the subtypes of necrosis. And so we're going to actually spend a good degree of time on the subtypes of necrosis. The first type of necrosis is called coagulative necrosis. What is coagulative necrosis? It is necrotic tissue that remains firm, i.e. the tissue sort of coagulates, or why don't we say the cellular proteins actually coagulate so that the cells retain their shape and the organ structure is preserved. So otherwise stated, the tissue dies. However, the general structure of the tissue remains, and if we were to look at the tissue on a microscopic slide, we would see that the cells are present and the architecture of the cells is present. However, the nuclei have disappeared. And so that's what we say here. It's the tissue remains firm. There's coagulation of cellular proteins. However, the nucleus disappears. The characteristic circumstance in which coagulative necrosis occurs is ischemic infarction. Every organ in the body that undergoes ischemic infarction will actually exhibit coagulative necrosis. The only exception is the brain. So, again, coagulative necrosis is seen with ischemia of any organ except the brain. Here's an image from your text, figure 1.5, that just highlights what we mean by coagulative necrosis. Now, let's start with this image here on the right. This is the glomerulus, what I'm circling, and these are the tubules. And you can clearly see that the glomerulus contains nuclei within the cells, and that the tubules also contain nuclei within the cells, and that's normal. I just put this normal picture here to establish a baseline in your mind. Now let's look here on the left at the necrosis. Here's the glomerulus. You still recognize that it looks sort of like a glomerulus. However, there are no nuclei within the cells. Here are the tubules. Still recognize that these are tubules. However, there are no nuclei, no blue nuclei within these cells. And so this histologic picture is characteristic of coagulative necrosis. A couple important derivative points regarding coagulative necrosis. Usually, the area of infarcted tissue is wedge-shaped and pale. All right. Usually, the area of infarcted tissue is wedge-shaped and pale. This is not so hard to remember. Basically, this is based on a principle, an anatomic principle, and that is that, generally speaking, when vessels enter into an organ and feed an organ, they dichotomously branch as they go through the organ. So let's say that there is a vessel coming this way, and then there's a branch point here, and then another branch point here as the artery branches to feed the organ. If you cut the blood supply here, everything from this vessel and everything from this vessel is going to die, and it's going to create sort of everything in the distribution of this area will become what, like a wedge-shaped, notice that this looks like a wedge-shaped area of necrosis. And one of the classic features to be aware of is that the wedge points to the area of occlusion. And that's because it's pointing to the area where the dichotomous branching was occurring. So I don't think it's hard to remember that these areas are usually wedge-shaped. Why are they pale? Well, same principle. I mean, if this is the blood supply coming in and this is the blood supply branching, then you cut the blood supply. Once you cut the blood supply, this tissue is going to die. And once it dies, blood will obviously not be able to come back into it, so the dead tissue will look pale. No rocket science, pretty straightforward. Here's a picture from your text, figure 1.5, and it's basically highlighting that this is the cortex of the kidney, and this area here represents the area of infarction. There was probably an occlusion here, and this tissue within the area of the triangle has died. Now, you can partly recognize by the fact that there is indentation of the tissue here, but also it's a little subtle, but there's disruption of the normal tissue in this region. And so this is a classic example. It's relatively pale, and it's kind of wedge-shaped. Gives you an idea of what we're referring to. Another important principle, or derivative principle, is that we could get red infarction within a tissue. And red infarction requires two things in order for it to be seen. First, blood must re-enter the tissue. And second, the tissue must be loosely organized so that it can hold the blood, giving it a red color. So these two things are required. Blood must re-enter the tissue, and the tissue must be loosely organized. A classic example is shown here in your text, and this is figure 1.6. This is a testicle, and this testicle has undergone hemorrhagic infarction. Basically, the idea here is that this testicle is completely red. Now, in order to understand this, we probably should take a step back and remind ourselves that coming in this area here, maybe I should create a blank screen to make it more easy to see. When we think about the testicle, coming into the testicle, you've got this cord which basically the testicle hangs on, and then this is the testicle here. Now in this region there is a thick-walled artery and there is a thin-walled vein. The classic mechanism by which testicular infarction occurs is that the testicle twists on this cord. And when it twists on the cord, the artery continues to allow blood to flow in because it's thick-walled. However, the vein collapses. And when the vein collapses, blood comes in, but blood can't go out. And if you recall from our lecture concerning ischemia, one of the ways by which you can get ischemia is blocking of the venous supply because fresh blood would not be then passing across the organ. So the organ will die because the vein, or the tissue will die because the vein is blocked, and blood will continue to re-enter because the artery remains somewhat open relative to the blockage of the vein. As blood piles up within the testicle is relatively loose, the tissue of the testicle is relatively loose, and that will then create what we call a red infarction. And it is important for examinations to be familiar with the distinction between a pale infarction and a red infarction. Okay, so let's continue on. The next type of necrosis is liquefactive necrosis. And guess what? In liquefactive necrosis, the tissue becomes liquefied. So we've got necrotic tissue, or dead tissue, that becomes liquefied. Now, the way by which the tissue becomes liquefied is that there is enzymatic lysis of the cells. The cells get acted on by some enzyme, and that is what basically destroys the tissue, resulting in the liquefaction. It is seen characteristically in three circumstances. The first is brain infarction. Recall that I told you that when you cut the blood supply to any organ in the body, you actually get coagulative necrosis. However, if the blood supply is cut to the brain, that would result in liquefactive necrosis. So that would be one important circumstance that results in liquefactive necrosis. One of the questions we can ask ourselves is, why is it that within the brain we get liquefactive necrosis, but in all other tissues we get coagulative necrosis? And the answer is because the brain contains microglial cells, which are derivatives of monocytes. They're sort of the macrophages of the brain. And the microglial cells contain hydrolytic enzymes that trash the tissue or destroy the tissue after it dies. And so this is mediated, this circumstance here is mediated by microglial cells, and that's actually an important principle as well. The second circumstance in which we might see liquefactive necrosis is in an abscess. Now, an abscess is a walled-off area of dead tissue, and it becomes liquefied because the neutrophils contain hydrolytic enzymes, and those hydrolytic enzymes, they destroy the tissue, resulting in the liquefaction of that tissue. And so if I asked you what mediates liquefactive necrosis in the case of an abscess, the answer is the enzymes from the neutrophils. The third circumstance that we might see liquefactive necrosis is in pancreatitis. What is pancreatitis? Remember that pancreatitis is a disease process in which we activate the enzymes of the pancreas within the pancreas itself. Now, normally the enzymes of the pancreas would obviously get activated in the gut and digest food. However, if the enzymes of the pancreas get activated within the pancreatic parenchyma itself, the pancreatic enzymes will digest the pancreas. And it's the digestion of the pancreas that results in a liquefactive necrosis of the pancreas itself. Now, I keep emphasizing this pancreas itself because in pancreatitis, we also see another type of necrosis of the surrounding fat, the fat that surrounds the pancreas, but that's called fat necrosis, and we'll talk about that in a minute. So actually, pancreatitis kind of has two types of necrosis associated with it, both liquefactive, of the pancreatic parenchyma, and fat necrosis of the peripancreatic fat. And if I asked you what's the mechanism on an examination of liquefactive necrosis in pancreatitis, the answer is that it's the enzymes from the pancreas that are digesting the pancreatic parenchyma.